A very good morning, friends. Today we will discuss about an important topic, an important short note in biochemistry, that is HMP shunt. HMP shunt means Hexose MonoPhosphate shunt. So these are the other names for HMP shunt. First one is the pentose phosphate pathway. In this pathway, there is generation of number of pentose sugar. That's why it is known as pentose phosphate pathway. This is an alternative pathway for oxidation of glucose molecule. This is an alternative pathway to glycolysis and TCA cycle. Glucose undergoes oxidation, and in glycolysis and TCA cycle it will give CO2. So this is an alternative pathway or shunt pathway. That's why it is known as shunt pathway for oxidation of glucose, in which glucose or glucose 6-phosphate through this pathway gives you CO2. And it is also known as monophosphate because as we have already seen that in glycolysis there is generation of number of bisphosphates, but in this pathway there is generation of monophosphates. That's why it is known as Hexose MonoPhosphate shunt. This is also known as Dickens and Horeker pathway. Then phosphogluconate, it is an intermediate in this pathway. That's why it is also known as phosphogluconate oxidative pathway. There are two important steps in this pathway. First one is the oxidative step, and second one is the non-oxidative step. So oxidative step, it mainly occurs in the liver, adipose tissue, then adrenal cortex, testes, ovaries, lactating mammary gland, and erythrocytes. So liver, adipose tissue, and RBCs, these are the main site for the oxidative phase of HMP shunt. While non-oxidative pathway, it occurs in the all cells of the body. It deals with the synthesis of pentose sugars, and these pentose sugars are ultimately required for the DNA and RNA synthesis. So it occurs in the all cells of the body. So the starting molecule in the HMP shunt is the glucose 6-phosphate. So glucose 6-phosphate, it is converted into 6-phosphogluconolactone. This is a rate limiting step in this pathway. It is by the enzyme glucose 6-phosphate dehydrogenase. So this is the main and rate limiting enzyme in this pathway. It requires NADP. It requires NADP as a coenzyme, and NADP is converted into NADPH plus H ions. So this is the first rate limiting step in the HMP shunt. Then with the help of gluconolactone hydrolase, this 6-phosphogluconolactone, it is converted into 6-phosphogluconate. Then with the help of 6-phosphogluconate dehydrogenase, it is a NADP dependent reaction again. NADP, it is converted into NADPH and H ions, and there is liberation of CO2 through a intermediate, that is 3-keto-6-phosphogluconate. Through this intermediate, by decarboxylation, it is converted into ribulose 5-phosphate. So these steps in the HMP shunt, they are known as oxidative phase. and it is important in the generation of NADPH. So this NADPH, it is utilized for the reductive biosynthesis, that is, it is utilized for fatty acid synthesis, cholesterol synthesis and steroid synthesis. So this pathway, it is mainly in the liver and adipose tissue, occurs mainly in the liver and adipose tissue. Now coming to the non-oxidative phase of this pathway. In the non-oxidative phase, this ribulose 5-phosphate, it is epimerized with the help of epimerase. It gives xylulose 5-phosphate, and on isomerization, with the help of isomerase, it gives ribose 5-phosphate. This ribose 5-phosphate, it is precursor for the nucleotide synthesis, that is inosine monophosphate, which gives nucleotides. So it is important in DNA and RNA synthesis. Now with the help of transketolase reaction, which is thymine pyrophosphate dependent, and RBC transketolase level is measured to assess the thymine status of the body. So this is TPP dependent reaction, transketolase, in which there is transfer of 2 carbon from this xylulose 5-phosphate to the ribose 5-phosphate. It leads to the generation of pseudoheptulose 7-phosphate and glyceraldehyde 3-phosphate. Now with the help of transaldolase reaction, there is transfer of 3 carbon from pseudoheptulose 7-phosphate that leads to the generation of erythrose 4-phosphate and fructose 6-phosphate. This is a transaldolase reaction. Again, there is transketolase reaction in which this erythrose 4-phosphate, it condenses with xylulose 5-phosphate to form fructose 6-phosphate and glyceraldehyde 3-phosphate. There is formation of 2 molecules of glyceraldehyde 3-phosphate, which gives ultimately fructose 6-phosphate. If you start from the glucose 6-phosphate, there is utilization of 6 molecules of glucose 6-phosphate, and the first carbon from each glucose 6-phosphate, it is liberated as carbon dioxide, and 5 glucose 6-phosphates, they are regenerated in the HMP shunt. So there is liberation of 6 molecule of CO2 in the oxidative phase by oxidative decarboxylation, which is catalyzed by 6-phosphogluconate dehydrogenase. Then this fructose 6-phosphate, if you start from the beginning, there is involvement of 6 glucose 6-phosphate, which gives you 6 molecule of ribulose 5-phosphate, which gives you 6 molecule of ribulose 5-phosphate. Then this gives 2 molecule of ribose 5-phosphate and 4 molecule of xylulose 5-phosphate. Now this will give you 2 molecule of pseudoheptulose 7-phosphate and 2 molecule of glyceraldehyde 3-phosphate. there is 2 molecule of erythrose 4-phosphate and 2 molecule of fructose 6-phosphate. Now this erythrose 4-phosphate combines with the 2 molecule of xylulose 5-phosphate. It gives 2 molecule of fructose 6-phosphate and 2 molecule of glyceraldehyde 3-phosphate. So there is generation of, this is the first and second, here is the 2 plus 2 4 plus 1 5. So this 5 fructose 6-phosphates, they gives ultimately 5 molecule of glucose 6-phosphate. So there is regeneration of 5 molecule of glucose 6-phosphate. If you see the overall reaction, there is involvement of 6 molecule of glucose 6-phosphates plus 12 NADP plus 6 molecule of water. It will give you 5 molecule of glucose 6-phosphate plus 12 NADPH plus 12 H ions plus 6 CO2. So this is also known as oxidative pathway for glucose, and 10% of glucose, it is oxidized by this HMP shunt, and liver and RBCs utilize 30% of glucose through the HMP shunt. Now coming to the importance of HMP shunt. In the HMP shunt, you will get two important products. The first one is the NADPH and second one is the pentose sugar. So pentose sugar, it is utilized for the DNA and RNA synthesis. DNA, RNA synthesis, also utilized for the nucleotide synthesis. For example, ATP, it is also required for FAD, NAD, coenzyme A synthesis. Now coming to the uses of NADPH. HMP shunt, it is a source of NADPH. The another source is the malic enzyme. This is the main source of NADPH. It is required for the reductive biosynthesis of fatty acid, steroids and cholesterol. Then it has free radical scavenging action. For this action, you require superoxide dismutase, peroxidase and glutathione reductase. So it is important for the action of glutathione reductase, which is a NADPH dependent enzyme. Then it functions as an antioxidant by scavenging the free radicals, and it is required for the bactericidal action of macrophages. It is required for the bactericidal action of macrophages. Then third, it is important in maintenance of RBC membrane integrity. In the absence of this NADPH, or if the level is decreases, it leads to the hemolytic jaundice, because it is required for the production of reduced glutathione by glutathione reductase, which is a NADPH dependent enzyme. This reduced glutathione is responsible for maintenance of RBC membrane, and again this NADPH, it prevents the formation of methemoglobin. It keeps the iron in the ferrous state. So that's why it is important. Then it is important in maintaining the permeability, it is important in maintaining the transparency of lens. Transparency of lens. So in this way, NADPH, it has number of functions. It also required for the detoxification reaction, which is carried out by the cytochrome P450 enzyme system, which is a hydrolase. It requires NADPH for its action. Then coming to the clinical significance of this HMP shunt, means disorders or diseases which are related to the HMP shunt. The first important is the glucose 6-phosphate dehydrogenase deficiency, that is known as G6PD deficiency. As such, it is a asymptomatic condition, but when oxidant drugs are given, in the treatment, for example, primaquine or sulfur drugs, that is sulfamethoxazole. If these oxidants or some antipyretics, that is acetanilide, if these drugs are given in the treatment, they increases the production of free radicals, and as there is problem in G6PD dehydrogenase deficiency, it lead to the decrease in NADPH production, which is required for the antioxidant functions, and this free radical causes damage to the RBCs, and that leads to the hemolytic anemia or hemolytic jaundice. So increased destruction of RBC lead to the release of hemoglobin, and heme degradation leads to the increased bilirubin in serum, that leads to the jaundice. So this is the main regulatory enzyme of this pathway, that is glucose 6-phosphate dehydrogenase deficiency lead to the G6PD deficiency, which mainly after the administration of oxidant drugs, that is primaquine, sulfamethoxazole, favabins and acetanilides, that causes free radical formation, damages the membrane, lead to the hemolytic jaundice. Second important is the transketolase activity. Related to the transketolase activity, that is Wernicke's Korsakoff syndrome. This is in the alcoholic. In alcoholic, there is decreased absorption of thymine. Also, there is decreased conversion of thymine to thymine pyrophosphate. This is the active form of the thymine. That leads to the functional deficiency of thymine pyrophosphate, and this main reaction it affects the transketolase reaction, which is involved in the HMP shunt. So in this Wernicke's Korsakoff psychosis, there is different symptoms, that is the mnemonic is GOA MP, that is global confusion, ophthalmoplegia, ataxia, memory loss and psychosis. This is due to the decreased level of thymine pyrophosphate that affects mainly the pyruvate dehydrogenase complex, and that lead to the decrease in the synthesis of ATP. So this is all about the clinical significance of HMP shunt. Summarizing the all, this pathway is divided into two phases, oxidative and non-oxidative phase. It mainly occurs in the liver, adipose tissue and RBCs. It leads to the generation of NADPH and pentose sugar. Pentose sugar is a precursor for the nucleotide synthesis involved in the DNA and RNA synthesis. NADPH, it is mainly involved in the reductive biosynthesis of fatty acids, steroids and cholesterol.